we can see new amino acids in this capsid. In other words, we can see new antigenic profiles of this virus.
Hello, I'm Peter Best. It's my pleasure to welcome you to Meet the Expert, the series of podcasts on swine health management in practice brought to you by Boehringer Ingelheim. For this episode of Meet the Expert, our subject will be the porcine parvovirus, or PPV. And I'm very pleased to say the expert helping us is Professor André Felipe Streck. Professor Streck, hello and welcome.
Dear Peter, thank you for your kind invitation. I would like to thank the entire team of Boehringer Ingelheim for the productive dialogues that we have. And also it's an honor to be here, and I hope to pass on some interesting information.
Thank you. Let me tell our listeners, please, that your PhD, it's in veterinary sciences, isn't it? And you're now based in southern Brazil as Associate Professor in the Institute of Biotechnology at the University of Caxias do Sul. Has the study of the porcine parvovirus played a big part in your working life?
Yes, indeed. Well, after my graduation in the med vet school, I started to do a master degree with Professor Cláudio Canal in the University of Rio Grande do Sul. And in this time, PPV, porcine parvovirus, was a subject that had a growing interest at my university. I remember to discussing the findings of several mummifieds without a diagnostic at this time. And also in this time, that was the year 2006, we had the porcine circovirus too here in Brazil. That was a huge problem without a commercial vaccine. And in this point, we performed several autogenous vaccines. We could see some significant improvements with our vaccines. And in this entire scenario, PPV was a possible X factor also for circovirus. And with this, we have this interesting to study a little bit more PPV.
But does PPV still need to be investigated, Professor Streck? Because after all, it's more than 50 years since it was shown to cause SMEDI syndromes of stillbirth, mummification, embryonic death, infertility. And we've had parvovirus vaccines to use in our swine breeding herds for at least the last 40 years. Mm-hmm. What was there still to be investigated?
Yes. It needs to be more investigated. Thereafter, we started to perform some studies with Professor Uwe Truy in Leipzig, mainly focus on evolutionary aspects of the virus, and we can observe that the occurrence and distribution of new antigenic types of porcine parvovirus and new PPV vaccines that may induce a greater immune response and cover all the prevalence strains, in my opinion, are quite needed.
Yeah, but the key question then with these new strains would be whether the long-established PPV vaccines that I mentioned, whether they're still effective even when we're confronted with new strains.
Yes, I always say that vaccines are still effective, always, and we need to practice regular vaccination. But I believe that new studies can make an increase in their efficiency.
So we can continue to rely on the vaccines of the past, but we need to develop new ones for the emerging conditions.
Mm-hmm. Well, I believe the process of vaccinal improvement should be something continuous. since we see that the virus has also a continuous evolution, I also emphasize that the process of develop vaccines can take about 5 to 10 years. So it's important that companies always look out for improvements. That's very important for the market.
Yeah. At this stage, Professor, we're all familiar with PPV, as I say, over many years, but let's remind our listeners, please, how and where PPV infection is seen in practice. First, we must state, I suppose, that it causes maternal reproductive failure, especially in first parity gilts and second parity sows, would you say?
Yes, absolutely right. The only well-established clinical signs after a PPV infection is the maternal reproductive failure. Uh, Even after experimental inoculation, and I remember that in this experimental inoculation, we use huge doses of virus, and even so, adult animals they remain clinical healthy. Uh, Maybe we can observe a moderate lymphopenia and a little bit fever, but mostly is the reproductive failure the important sign. We can also observe some diarrhea and skin lesions. That was described for PPV infection. However, these events may represent some hair findings, some artifacts, and the etiologic role of the virus in some symptoms are not fully understood.
Yes, it could be a co-infection or so on. Yes.
Maybe,
Yeah.
yes.
I didn't refer to the other sign of delayed estrus, which many people talk to me about in connection with PPV. It's a very common sign in infected sows and gilts.
Mhm. Yes. The in thousand gills, the mostly the mummification is the most important sign. Uh, Mostly is the embryonic death followed by reabsorption of fluids and then soft tissues. Uh, that's the predominant sequel of PPV infection. Uh, this we can see also that several bacteria and other virus can perform the same thing, and this way is very important to perform a nice diagnosis, to use laboratory tests to perform this diagnosis.
I'm with you. But this mummification, it signifies the infection has occurred in the gilt or the sow before day 70 of gestation, I understand. After infection, you've got, and the fetus is up to about day 35, I think they'd probably be reabsorbed in many cases. So between day 35 and day 37, you've got the mummification, have you? And with PPV, the piglets may have become infected, died, and become mummified at different stages, can they? Not all at once from the
Mhm.
same uterus.
Okay. Well, the pathologic sequela caused by PPV is related to the gestational period in which infection usually occurs. Uh, at the gestational beginning, the entire conceptus is protected by the zona pellucida and therefore is resistant to infection. After that, in in the stage of embryo, infection can result in reabsorption, or in other words, embryonic death and thereafter reabsorption. From the day 35 on, we have the beginning of the ossification, and then the death of this fetus can result in mummification. Finally, when we have the 70 days on of gestation, the fetus already have some immune system and they may resist virus infection. Normally, an infection in this stage is quite controlled, and the piglet is born with antibodies. Well, about the infection in different fetus is quite right. Not all fetus are necessarily infected. Many times we have mummification in only a few fetus, and others they manage to survive the infections. It's important to say that what is the hallmark of porcine parvovirus are litters with dead fetus from various size, including mummified ones, and along with stillborns, healthy pigs in firstly parity guilds. That's the hallmark, the big hallmark of the virus.
Yes, the piglets, by the way, that are immunocompetent because they've been infected after day 70, when they're born, they're alive, but are they showing any clinical signs of weakness or paleness or anything like that? Or do they look quite normal?
Mostly they look normal. They can be a little bit weak and probably more uh, have a more propension to another disease like Escherichia coli or rotavirus, but mostly they are quite normal.
Yes. Now, earlier I referred to parvo as a SMEDI virus, and typically SMEDI is by definition without abortions, but I've seen occasional references to abortions. Uh, would it be that either abortions or recurring estrus are in fact quite common, or are they very rare?
PPV can make some abortions, but I highlight that mummification is the most classical signs of the virus. But yes, we can have some abortions. For other side, some pathologists use the term abortions to define some virus signs of reproduction failure. For example, also for mummification. And this way, this can cause a misunderstanding of the term abortion also.
I understand, yes. So let's go back to diagnosis. You were starting on that. Have there been significant developments in PPV diagnosis in recent years?
Yeah. Well, the diagnosis of PPV started with normal hemagglutination techniques, and thereafter with virus isolation. These both techniques, they are not so much sensitive, and usually we can reach a lot of false negative results with these techniques. Thereafter, in the 90s, we started to perform PCR for PPV that helped us a lot to discover new strains, to see that we had, yes, PPV in our hearts. And recently, we started to perform some real-time PCRs for PPV. That's real-time PCRs for PPV, in my opinion, are very important to see also the amount of DNA of this virus. And in this technique, we can observe it PPV was maybe some laboratory artifact, was some secondary cause, or was the primary cause of this mummification.
And would you say then that, as far as diagnosis is concerned, we've improved both in our techniques? We know which samples we should be sending to the lab for diagnosis, and we're getting a reliable result back. Would that be true, or would that be too strong?
Well, no, it will be true. It's quite important to remember that for PPV, we had a lot of autolysis material, and when we send samples to the lab, the conservation of the samples is very important, and also the sensitivity of the technique is also important to reach this DNA. Without a sensitive technique, without a correct conservation of this material, we cannot reach the diagnostic. In this point, it's also important to remember that maybe in the future we can talk a little bit more about new diagnostic techniques, like point of care techniques, that will be very nice to perform some diagnostics in the field also, and will be very important for PPV also.
So a pen-side test would be a possibility.
Yes, I think that will be very important uh, to to see if the abortion was, or sorry, the
Yes.
mummification was caused. I say abortion because the pathologists of my university, they told us, well, we say here abortion to all reproductive failures, and I usually say abortions for porcine parvovirus, but I will talk mummifications.
Yes.
And this way, it will be very important to perform this test in the field to see if the mummification was caused by parvovirus, porcine serovirus, or other agent there.
Right, I'm with you. Just let me take a moment, please, to say
Yes.
to our listeners, thank you for joining us. You're listening to a Meet the Expert podcast brought to you by Boehringer Ingelheim, and we welcome feedback from you on all of our podcasts. More episodes of the Meet the Expert series will be made available to you soon, once again featuring an array of swine health experts in various countries, giving you the latest information on the important pathogens that affect pigs. So thank you for joining us today, and please stay tuned for future podcasts. So back to you then, Professor Streck. PPV is a DNA virus, isn't it? And yet, from your remarks, it's changed genetically quite a lot since it was first reported. When was that? In the mid '60s, I think. So there have been genetic changes even in this virus.
Yes, it's a DNA virus. However, we can estimate for PPV and also for another parvovirus, for example, canine parvovirus, um, a very high mutation rate or substitution rate, mainly in the viral protein gene. That means in the external capsid, and that's the part of the virus that have contact with the immune system. This way is very important to study the evolution of this virus, and to understand that we have a process, an evolutionary process happen in this virus.
I'm with you. But you mean it was, I described it as a very stable virus, but it was evolving all the time. It wasn't
Mhm.
as stable as we might have imagined.
Yes, we can observe that by applying some analysis, for example, molecular clock. that the main diversions of this virus were introduced in the last 40 years. Most importantly, in recent years, we can see new amino acids in this capsid. In other words, we can see new antigenic profiles of this virus. And these antigenic profiles were observed in strains from several countries, mostly in Europe. And importantly, these evolutionary hotspots were found just in the surface. In other words, located points located with huge contact with the immune system of the host.
So exactly at that point of contact, you get these viral protein changes which affect the surface of the capsid, what I still call a protein shell of the virus. I don't know whether you'd like that description, but anyway, that's how I like to think of it, a capsid
Of
in that
course.
way. But you said it's been going on 60 years or something, did you say? I mean, this genetic divergence that we see today has its roots going back that far.
Yes, we can perform this studies with the genetic variety that we have today, but it's important to say that if we have more samples, we can reach a better precision in this studies. And for this reason, it's better we need to do some new researches and get better answer. Probably the virus, the evolutionary process of the virus will be going on for decades, but our information is located mainly in the last 50 years, 50 or 60 years, unfortunately.
And this evolution was by mutation. There wasn't any recombination of virus involved in that.
No, no. In our studies, we try to see some recombination. We try to see some recombination signal, and we can observe a greater signal. There is small signals of recombination, but I really don't believe that parvovirus make some improvement in their genetic diversity by recombination. I think the what drive this evolutionary process of the parvovirus are points of mutation.
Yeah, I'm with you. But yet, until 20 years ago, we were thinking this was a virus that hardly changed at all. I mean, there was a sort of apparently a false assumption that this parvovirus did not mutate. Why was that assumed then?
Well, parvovirus use host polymerase for replication. We have an DNA polymerase for our DNA replication, and our eukaryotic cells have this polymerase complex that have a high confidence in the generation of new DNA copies, and that's called a repair unit. And they have a very, very, very low substitution rate. And this is also good, because if you don't have this repair unit, we will have a lot of cases of cancer in the animals and humans. That's nice. However, or therefore, it makes sense that parvovirus were considered more stable than another virus, and it was considered to have a substitution rate or mutational rate close to their host, the pig. In contrast, RNA virus, which have their own RNA polymerase, without this repair unit, they have a very, very aggressive substitution rate or mutational rate.
So the copies were very close to the original with the parvo, and less so with the RNA. You get copies which were quite different.
Yes, and what we see in the last studies is that, on the contrary, porcine parvovirus use the host polymerase, but they, for some reason, the repair unit don't work so well with a virus. Usually, in other words, this DNA polymerase works like a normal, a usual polymerase of an RNA virus, and insert a lot of mutations in this DNA sequence.
Does this help to explain why PPV has received relatively little attention from a research perspective?
Yes, but I also believe that the vaccine was very effective in the 80s. Uh, Another reason is that the diagnostic techniques available at this time were not so sensitive in the past, and it was much more difficult to get the diagnosis, to reach the diagnosis of mummified fetus. And it's important to say that the molecular techniques available today, they have greater sensitive compared to other techniques. I think these three reasons are maybe the, the great reasons to think that PPV was not a huge problem at this time.
Right, I'm with you. There seems, of course, since the year 2000, to have been almost a cascade of new PPV findings in different countries. What does that process of evolution produce today then? We have multiple strains and genotypes in different countries. What are we facing today?
Well, yes. The first study observing some genetic variability of PPV from field strains came from Brazil, from sequence isolated in the 90s, and they observed a lot of new mutations, mainly in this virus shell or capsid. And they observed also two main phylogenetic groups. Subsequently, we have these studies from Professor Truyen in Germany, and they also observe two other phylogenetic clusters in there with the 27A, with the already I started a predominance of the 27A strains, and thereafter there was a lot of studies observing new clusters, observing 27A like strains, and in other countries, in other continents, like Austria, China, Romania, and so on. Uh, Importantly, we observe a predominance of some strains in Europe in these times, and in other parts of the world in these times.
Was this predictable or surprising that such an event would take place in different parts of the world and produce apparently similar viruses?
No, in my opinion, not so that was not so surprising because the swine industry is quite related. The genetic of the pigs, they came from 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 some countries, and probably the virus pass also. We export virus also with the genetic of the pigs.
And what's different about these strains called 27A that originally were isolated in Germany, as you say? Does 27A differ both genetically and antigenically?
Yes, 27A, this strain, and I like to say today that we have 27A like strains because the original 27A maybe just don't exist anymore. They have some new amino acids in the capsid, and these amino acids are located in a very, very important part of this capsid, in a part that we call loops. And in these loops, or better, these loops are located in epitopes that are just the parts that stimulate the human systems. And also with new research, we can observe that these epitopes, or sorry, these changes in some points of these loops can have some another effects, like a better replication activity of these strains. And this way, the 27A like strains are very important today in the field, and
Uh,
probably predominant.
yeah. So they have antigenic differences. Is 27A in general more virulent?
That's a nice question. Yes, Professor Uwe Truyen performed some studies, and we can see in these studies that 27A is more virulent, yes, compared to standard sequence. And importantly, they can reach a better replication for in some cases, and also have a reduced neutralizing or better also antibodies raised against some older strains, have some reduced capability against these 27A strains.
So again, vaccine would it be older vaccines would be making antibodies which may not be so effective against 27A.
Yes, but I need to remember that for a vaccination, that's another factors that play a role. For example, we have the technology of the vaccine with the technology of the adjuvant, and we have the also another technologies. For example, when we perform some protein purification, we can reach a better doses, a better quantity of protein, and that's quite important also in the vaccine. And when we talk about vaccine technology, uh, the genetic characteristic of the base vaccine strain is important, but we need to remember that are another factors in a vaccine as well.
Thank you. Now I hope we will continue this conversation, the implications of vaccination in our second conversation, a second podcast, Professor Streck. But I regret for the moment I have to say thank you very much because we've come to the end of this podcast. And may I remind our listeners, we've been talking in Brazil to Professor André Felipe Streck, at the University of Caxias to Seoul, and we've been talking about the porcine parvovirus, or PPV. Thank you for joining us, Professor Streck, and to our listeners. Please stay tuned for a further conversation. Goodbye.